Stem to Stern, the video panel series from the Pharma Navigator, where expert panels will delve into pertinent industry topics in more detail. During each of these peer-to-peer forums, we'll navigate the full life cycle of biopharmaceutical innovation. Hi everyone, thanks for joining me for this next instalment from Stem to Stern. I'm Felicity Thomas and I'm the Associate Director of Editorial and Content for the Pharma Navigator, and I will be the host for today's discussion. Today we're going to be focusing on oral peptides, and in this episode I'm joined by Bobby Cicento, Trevor Williams and Zachary Morseth, who are all from Alcami. Thanks very much for joining me today, Bobby, Trevor and Zach. Could you please provide us with a brief introduction to yourselves for our viewers? And Bobby, we'll start with you.
Sure. Thank you for the introduction. My name is Bobby Cicento. I'm a senior scientist working in Alcami's Formulation Development Laboratory, located in Wilmington, North Carolina. I've been with Alcami almost 5 years now, and in the industry, mainly focused on oral formulation development for 11 years.
Zach?
So my name is Zach Morseth, and I'm a manager within our bioseparations and mass spectrometry group here at Alcami. My team focuses on method development and validation for a range of biologic methods, like things like liquid chromatography, capillary isoelectric focusing, and capillary electrophoresis, along with extended characterization techniques like SEC-MALS, LCMS, analytical ultracentrifugation, and circular dichroism.
Trevor?
Hi, my name is Trevor Williams. I'm also a senior scientist at Alcami. I work very closely with Bobby. We're the two formulation scientists for our group. I've worked for Alcami now for about I think it's gonna be actually 4 years coming up now. And I've been in formulations for about 5 years. I did some analytical work for my first 10 year when I worked at Bayer back in 2016 I started in Bayer, so it was about 5 years there as well. So at Alcami now, I work in the formulation development, but I also work in a lot of the early analytical developments for a lot of these early stage drugs. So very interesting to have this conversation today, and thank you for having us on. Really appreciate it.
As mentioned before, we're here today to talk about oral peptides. Now, thanks to effectiveness and biocompatibility, peptide therapeutics have the potential to become blockbusters on a scale that, we previously saw within the monoclonal antibody space. However, peptides also present development challenges, particularly for oral delivery, as they're susceptible to hydrolysis, enzymatic degradation, and they also have poor bioavailability. So, Bobby, I'm going to ask you, please, could you run through the potential strategies that are available to overcome these obstacles to formulating a broader range of oral peptide therapies?
Okay, yeah. So from when we see peptides coming in for oral formulation development, one way to stabilize them from a molecular biology standpoint is actually small substitutions on the peptide itself. So to simplify that, a peptide that is used for injection might be slightly different. The version of it might be slightly different in an oral format. And that's simply just, as I mentioned, molecular modification such as pegylation, cyclization, and lipidization. So that's basically different groups added to the peptide chain. Now, from a delivery standpoint, one kind of umbrella strategy that we see with peptides is targeted delivery. So to prevent hydrolysis in the stomach, for example, acid hydrolysis, many peptides typically undergo an enteric coating process, whether that be in tablet form or an enteric capsule delivery. So those are two really strong ways to stabilize peptides for oral delivery.
Absolutely. And I'm going to just stick with you for a minute, Bobby, and I'm going to focus on permeation enhancers. These need to dissolve in a coordinated way along with the peptide in order to work. How can developers optimize a matrix to achieve such synchronicity?
Sure. So one way to kind of increase the bioavailability of peptides is the use of lipids. And that's been historically done in the soft gel industry. But there's many ways that that can be incorporated into our oral formulation. So that could be as simple as a single lipid system that increases intestinal permeation. That an example of that might just be simply MCT oil, um, dissolving your peptide in MCT oil and then somehow incorporating that into your oral formulation, whether that be a tablet or a capsule. Another strong way to increase bioavailability is take that one step further and incorporate a SED system. So that would be with a surfactant, co-solvent, and some other lipid. An example of that, a common SED system that we use might be, and again, building on the MCT oil, that could be your oil component, maybe a polysorbate as a surfactant, and a co-solvent like PEG or any version of the PEGs. And that would be a simple SED system that would increase the bioavailability of a peptide. Now, as far as evaluating the effectiveness of each of those formulation modifications, what we see is really heavy in vitro dissolution testing. Typically, that would include biorelevant medias such as FASF, FESF, and the FAST-GF medias to evaluate those single lipid modifications or a SED system. Typically, if we're formulating down that route, there may be different variations of the SED composition, as well as different oils evaluated. So that's kind of how the formulations are rank ordered and benchmarked. Another common permeation enhancer for peptides is SNAC, and that's the full name of SNAC is salcoprosate sodium, and that's commonly used in the Ribelsis formulation to increase the permeation enhancement of semaglutide.
Yeah, but with obviously oral peptides don't only pose, formulation challenges. They're very fragile and, that kind of molecular fragility isn't compatible with traditional oral solid dosage unit operations. Therefore, what process adaptations or alternative manufacturing pathways, such as continuous direct compression or specialized fluid bed processing, are necessary to handle peptides? Trevor, I'm going to hand over to you for this one.
Yeah. So obviously, in most oral solid dosage, we work with very small molecules. They're pretty rugged. They can go under a lot of stress. Oxidation is not really as much of a problem with these. So the main thing with these peptides is they are very structurally complex, and they're very sensitive to the environment that you could either add from adding moisture, light, sensitivity, mechanical stress. So usually, when we work with smaller molecules, we'll do wet granulation, we'll do prolonged blending, we'll even do some high shear mixing, we'll also do roller compaction. But with peptides, that's exposing them to a lot of these moisture, elevated temperatures, some high shear force, and even could have these oxidation reactions happen. So the one thing we really are looking for here is we're trying to really monitor and make a more peptide-friendly environment for when we're manufacturing. So like you said, one of the ways we can do that is we have seen continuous direct compression become a very important matrix. So with this case, you're kind of feeding all your ingredients in a very closed system. So when you are doing this continuous manufacturing, you're really allowed to have a much more sensitive environment and be able to really control your environmental controls and expose the peptides to much less outside forces. Also, when you're doing this, you're basically feeding all of your excipients and your ingredients in a single process, and then it's directly going into tablet compression. This also has a lot less mechanical force involved since you are not really mixing it for prolonged times. You're not stirring it with high mechanical force as well. So you're able to really make sure you can preserve the peptide. The strategy is also really good because it kind of gives you a much lower amount of API needed. You're making a much smaller batch here, which reduces the time the peptides are exposed, and also makes it so if there is a problem with the batch, you're not wasting a large amount of money here, because peptides are very expensive, and we are trying to introduce these fewer processing steps and also help make sure we're not wasting the peptides if something goes wrong during the batch. And then another innovation here would also be just because of we're seeing a difference in the way fluid beds are being made. They're becoming a lot more controlled, whereas traditional fluid beds, there's a very high amount of force that you're adding into it from a shearing perspective. You're also adding a lot of heat when you are drying it. Kind of like Bobby said, one of those ways is that we are adding the lipid granulations, which when you do lipid granulation, you really don't need to introduce heat because you're not adding water into the system. So we're getting a lot more of these controlled modern fluid bed systems that are designed to be a lot less intensive on the peptide itself. So we're able to kind of still have that granulation or uniform coating process without adding much mechanical strength. And also what really we need to work on here is just making sure that the manufacturing sites themselves have a lot of environmental controls set up. And this can be done by having multiple digital manufacturing technologies that can actually do your analytical testing while you're running the batches. So there's many tools out there that can give you blend uniformity, it can give you moisture content, it can give you tablet weight and compression force in real time. So instead of having to rely on external forces outside of it, you can kind of do this at the same time and make sure that your batch throughout the whole process is showing the results that you want. So really just the way you're going to see this is I think that the new formulation approach is going to be, even from the R&D stage, you're going to see a lot of these continuous manufacturing processes be introduced rather than in older times where we would basically make the formulation and scale up and then think about scale up once we got a good formulation. I feel like at this point now we really need to focus on the manufacturing process from the get-go so that when we do scale it up, we can reduce the troubles that we would see in scalability.
Additionally, analytical testing for oral solid peptides is complex because of the degradation products, which can be subtle and easily masked by dense excipient matrices or permeation enhancers. How are analytical and stability testing workflows evolving to ensure precise quantitation during real-time release testing and long-term stability studies? Zach, could you weigh in here, please?
Yeah, so peptides are already complex molecules, but when you put them into a solid dosage form, you're going to add a whole layer of excipients and permeation enhancers that are designed to help the peptide survive the GI tract and get absorbed. Now, the problem is those same excipients can interfere with the degradation products that we're trying to monitor. So a subtle change like a peptide that gets oxidized or maybe loses in a midgroup can get buried within that formulation matrix. Now, when it comes to analytical techniques, ultra high performance or UPLC remains our primary quantitative workhorse. So first, we start with chemical separation, and we're moving toward UPLC with this sub 2 micron particle columns, where these smaller column particles mean sharper peaks and better resolution. So these degradation products that used to collude with the excipient peaks can now be separated and quantified. We also look at column chemistry, and we know that not every column is created equal. So we're experimenting with core shell particles and alternative phases to find the selectivity that actually distinguishes our peptide degradants from the surrounding matrix. Now, alongside this UPLC, there are a few complementary methods that we also use. So UV-vis spectroscopy that gives us a fast orthogonal check on the assay value. It's not as selective as UPLC since it can't distinguish intact peptide from certain degradants, but it's useful during method development or even in-process testing. We also have circular dichroism, and that covers the secondary or higher order structure. So peptides can generally pass these purity checks when we do liquid chromatography, but they can still have a shifted secondary structure. So CD gives us that missing piece by confirming the molecule's conformation hasn't changed during these stability studies. And then when UPLC shows peaks that we can't identify, that's where mass spectrometry comes in. So with LC-MS-MS, we do identity confirmation and we pinpoint exactly where a degradation event happened, like a specific oxidation or a deamidation site. And then lastly, dissolution testing gives us that release behavior context. So combined with UPLC quantitation, we can see these dissolved fractions and tell us whether the peptide is releasing on the timeline that the formulation was designed for. So underneath all of this, we're doing force degradation studies to stress our methods with heat, light, oxidation, and even pH extremes to confirm that our method can detect solid state relevant degradants before we trust it for release or stability testing. So together, these methods and techniques really let us trust what the data is telling us, even when the formulation is doing its best to hide it.
So looking ahead, converting a highly predictable 100% bioavailable injectable peptide into a robust oral solid dosage form remains challenging. As the industry advances, should the primary benchmark for success be pushing the envelope to achieve higher absolute bioavailability? Or should there be acceptance of a low bioavailability and a focus on engineering technologies that minimize patient variability instead? Trevor, can you share your thoughts here?
So really here, the way I'm looking at it is you're looking at, are you trying to get a more optimized formula, or are you trying to show very good clinical relevant results with your formulation? Historically, peptides really have been pretty highly available. The formulations have been focused on getting the most bioavailability, but that's because it was in an injectable. You inject the peptide, you're really not seeing any major issues within the body system and no systematic issues to happen when it is being absorbed in the body. The problem is that when you do move into the oral solid doses, there's a lot more factors, like Bobby was kind of saying, with where it goes through the stomach, and you have to make sure it's going through all the different processes and organs in the body. So that's why at this case, you need to kind of look at it in a different way. I would say from the way we would like to do it is a more predictable model is much better than a more variable model. So you're going to see this if from a clinical standpoint, you'd rather have a drug that is consistently giving 3 to 5% in about all patients rather than a drug that has a very high absorbance in some patients and a very low in other patients. Because when we are looking at clinical models and patient safety, you really want to make sure you're giving a very predictable amount of this bioavailability. In that case, really, I think in the beginning, we should really focus on making sure that we have a drug that is very predictable, gives a solid bioavailability. But really, you have to look at these kind of in two parallel questions, because as technologies get better and as we move forward with learning more about the peptides themselves, I see both of these fields going in the right direction. You're going to see the formulations be optimized. We're going to be able to get more excipients that are more peptide friendly. We're going to have better engineering around the peptide molecules themselves. So that should also increase your bioavailability and also should increase your accuracy. But in my opinion, I think from a patient safety perspective, I'd rather have a drug that's giving 3 to 5% in everyone, and it's very safe, reproducible, and safe, rather than a drug that's very variable and where you're seeing sometimes 10%, but then 1% exposure in someone else. So I think that's really what we need to work here, is just making sure we're delivering a clinically consistent amount of peptide to everyone and making sure the patients are safe throughout the process.
Thanks again to the expert panelists participating in this episode of From STEM to Stern, Bobby Cizento, Trevor Williams and Zachary Morseth. I hope you enjoyed this episode, and I'll look forward to welcoming you to the next instalment of our panel discussion series. You've been watching From STEM to Stern from the Pharma Navigator, the essential video panel series for drug development and manufacturing professionals. If you've enjoyed today's discussion, be sure to subscribe to our YouTube channel, and you can also visit thepharmanavigator.com for even more expert analysis. Join us next time on From STEM to Stern.